Welcome to GIST Biomomed for your educational videos. Cell identity, specification, determination, and differentiation. In developmental biology, cell specification refers to the process by which cells are directed to specific fates of becoming specialized to perform particular functions within an organism. This process is crucial for the development of multicellular organisms, as it ensures that different cell types form in the right locations and at the right times. When a cell have not yet developed into a specialized cell, is said to be undifferentiated. Examples are stem cell and the totipotent stem cells in the zygote. Totipotent stem cell has the ability to develop into any cell type in the body. Some of the questions that are being answered here include, if genomes are equivalent, then how are all undifferentiated cells in the body containing the same gene that is derived from one zygote become different from one another? Or how does an embryonic cell acquire a trajectory towards a particular cell type? For cells to become different from one another, they have to individually become committed to particular cell fate before they finally become specialized. As the cell proceeds through the stages of cell fate identity, cell division ceases, and also, the cell phenotype may not look different from neighboring cells at the early stages of the levels of commitment. This is especially noticeable in embryogenesis. If you haven't seen our video on early embryogenesis, that described the morula and blastula stages of early embryogenesis. You can pause this specifying cell identity video and click on the link in the bio to watch the video. In our early embryogenesis video, we describe the morula and blastula formation that is mentioned in this video. Let's return back to the specifying cell identity lesson. When a cell becomes specialized, it is said to have undergone differentiation and referred to as a differentiated cell. The process of becoming committed to a specific cell identity can be divided into two stages: specification followed by determination. The early stage of the levels of commitment is the specification stage. In this stage, a cell fate is said to be specified when it differentiates by itself or autonomously in an environment that is neutral with respect to its developmental pathway, as seen in embryogenesis. In the diagram, two differently committed cells from the morula to blastula stage of early embryogenesis, presently at the specification stage of the levels of cell fate commitment, were isolated and placed in a petri dish. These petri dishes are neutral environments with respect to their development pathway, such as being with other morula-blastula cells. If the cell in dish A1 is fated to become a muscle cell, as shown in dish A2, and the cell in dish B1 fated to become a neuron cell, as seen in dish B2, in this specification stage, because they are in a neutral environment in the petri dish, both cells in the dishes, A1 and B1, will autonomously become muscle cell and neuron cell respectively as fated. However, at the specification stage, commitment to a cell fate, or identity, is capable of being altered to a different identity than its initial original fate. This is to say that the cell identity at specification stage is labile, or capable of change, or reversible. This diagram shows the same cell, specified to become a muscle cell, but this time it is placed in a population of other cells, specified to become neuronal cells. The fate of the transplanted muscle cell is altered by its interaction with its new neighbors, and it became a neuronal cell like them, instead of a muscle cell, because at the specification stage, a cell can adopt its identity based on the interaction with its neighbors, and the muscle cell has adopted the identity of the neuronal cell based on the interaction with the other neuronal cells in the dish. This shows the labial ability of cells at the specification stage. Therefore, at the specification stage, external influences matters because it changes the cell fate. Remember that the specification stage is the earliest key stages of the levels of commitment. The second key stage of the levels of cell fate commitment is the determination stage. This stage is an advanced step in the levels of commitment. A cell at this stage is capable of differentiating autonomously when placed alone in a dish, or even when placed in a cluster of other differently specified cells in a petri dish. A cell at determination stage will always be true to its specific cell fate. It is not affected by external influences. Here, we place the same muscle cell in the diagram, in a cluster of neuronal cell. But this time, because the muscle cell is at the determination stage, the presence of the clusters of neuronal cells surrounding it doesn't affect its fate to becoming a muscle cell, because it has moved further in level of commitment, and it is now determined to become a muscle cell at its present level of commitment, which is the determination stage. At this determination stage, it is assumed that the fate of a cell is irreversible. In summary, an undifferentiated cell, like those present during embryogenesis, mature through levels of cell fate commitment that cumulatively commit the undifferentiated cell to a specific cell fate. The levels of cell fate's commitment are, first specification, followed by determination, and finally differentiation. When an undifferentiated cell, like the stem cell, becomes specialized, it is said to have differentiated. The differentiated cell acquires specialized structural and functional elements that govern their own properties. A stem cell can differentiate into neurons, epithelial cells such as enterocytes, hepatocytes, muscle cells, connective tissue cell such as fibroblast, and many other specialized cells. A differentiated cell is naturally expected to maintain its functional identity for the rest of its life, except by some forms of unnatural course of interventions, like laboratory experimentations or disease states. During these interventions, gene expression patterns of the differentiated cell are altered. This type of interventions and their molecular mechanisms will be discussed in later videos. A differentiated cell is very different from an undifferentiated cell in many aspects. They are different morphologically, having different shapes and sizes. They are different in their differential gene expression patterns. The genes expressed in undifferentiated versus a differentiated cells are not similar. There are also differences in other genetic elements. In addition, differently fated differentiated cells are also different in their gene expression pattern, meaning the gene expression pattern of a muscle cell is different from a neuronal cell. The eventual proteins in the cells are also different. Understanding the concept of cell differentiation is also important in medicine. For example, medical scientists can translate the mechanism of cell differentiation into different stem cell therapies, where stem cells are used to repair damaged tissues or organs, in cancer treatment, and also in the treatment of genetic disorders. For more educational videos on specifying cell identity, please like, follow, and subscribe to this channel, and also subscribe to our social media website, gistbiomolemed.com. Thank you for watching and subscribing, and see you in our next video.